Today we are going to talk about one of the greatest scientific discoveries in human history, the discovery of the structure of DNA. This discovery, made in 1953, changed our understanding of life itself. It told us for the first time how living things store and pass on the information that makes them what they are.
I have heard of DNA before, but I am not sure I really understand what it is or why the discovery was so important.
That is a very common feeling. DNA is something most people have heard of, but the story of how it was discovered and what it means is far more dramatic and interesting than most people realize. It involves brilliant scientists, fierce competition, important contributions that were overlooked for decades, and questions about life that humanity had been asking since the beginning of recorded history.
And it also has consequences for medicine and science today, right?
Enormous consequences that are still unfolding. The discovery of DNA structure opened a door that scientists are still walking through. Everything from cancer treatment to genetic testing to the editing of genes in living organisms flows from what was discovered in 1953. Today, we will follow the whole story, from the earliest questions about heredity all the way to the present day. Let us begin.
So before scientists discovered the structure of DNA, what did they actually know about how living things pass on their characteristics to their offspring?
They knew that heredity existed, that children resemble their parents, that traits are passed from one generation to the next. The basic patterns of heredity had been described by a monk named Gregor Mendel in the 19th century through his experiments with pea plants. But what nobody understood was the physical mechanism. What substance inside the cell actually carried the genetic information?
And there were different theories about what that substance might be.
Yes, and for a long time the leading candidates were proteins, not DNA. Proteins are extraordinarily complex molecules that perform a vast range of functions in living cells. Scientists reasoned that because hereditary information is so complex, only something as complex as a protein could carry it. DNA, by contrast, seemed too simple and repetitive to contain the rich information needed to specify an entire living organism.
So scientists were looking in the wrong direction.
Many of them were, yes. But evidence began to accumulate that pointed toward DNA. The most important early experiment was conducted by a scientist named Oswald Avery and his colleagues in New York in the early 1940s. Avery was studying bacteria, specifically trying to understand what transformed one type of bacteria into another. Through careful and methodical experiments, he demonstrated that the transforming substance, the material that carried hereditary information from one bacterial cell to another, was DNA, not protein.
That sounds like a very clear result. Did scientists accept it immediately?
Surprisingly, no. Many prominent scientists resisted the conclusion for years. Some doubted the experimental methods. Others simply could not accept that something as seemingly simple as DNA could carry such complex information. Science does not always move in a straight line. Important discoveries are often resisted before they are accepted, particularly when they challenge deeply held assumptions.
You mentioned a book by a physicist called Schrödinger. How did a physicist contribute to this biological question?
This is a fascinating part of the story. In 1944, the Austrian physicist Erwin Schrödinger published a small book called What is Life? In it, he asked a profound question from a physicist's perspective. How does a living organism maintain its ordered complexity when the laws of physics suggest that everything should tend toward disorder? He proposed that genetic information must be stored in some kind of molecular structure, what he called an aperiodic crystal, that could encode a large amount of information in a stable form.
And this influenced other scientists.
Profoundly. The book was read by many young scientists and inspired several of them to turn their attention toward the question of the molecular basis of life. Both James Watson and Francis Crick, who would later discover the structure of DNA, cited Schrödinger's book as an important influence on their thinking. A physicist asking biological questions in a new way helped create the intellectual environment in which the discovery became possible.
So by the early 1950s, many scientists were working on this problem.
Yes, and there was genuine competition among several research groups. Groups in the United States, in Britain, and elsewhere were all pursuing the structure of DNA using different methods and approaches. The race was not formally organized, but everyone involved understood that whoever solved the structure first would have made one of the most important discoveries in the history of biology.
So who were the main people in this race to discover DNA structure?
There were several important groups. At Cambridge University in England, James Watson and Francis Crick were working together on the problem, building physical models of possible molecular structures. At King's College London, two researchers named Rosalind Franklin and Maurice Wilkins were using a technique called X-ray crystallography to study the physical structure of DNA directly. And in the United States, the famous chemist Linus Pauling was also working on the problem.
Can you explain what X-ray crystallography is?
X-ray crystallography is a technique that uses X-rays to reveal the structure of molecules. When X-rays are directed at a crystallized substance, they scatter in patterns that depend on the arrangement of atoms in the molecule. By analyzing those patterns very carefully, scientists can work out the three-dimensional structure of the molecule. It is an extremely powerful technique, but it requires great skill and patience to do well.
And Rosalind Franklin was particularly skilled at this technique.
She was one of the most accomplished X-ray crystallographers of her generation. Franklin had trained extensively in the technique and had a reputation for extraordinarily precise and careful experimental work. When she joined King's College London, she applied her skills to studying DNA and produced images of exceptional quality.
Tell me about Photo 51. What made it so important?
Photo 51 was an X-ray diffraction image of DNA that Franklin produced in 1952. It was a remarkable piece of scientific work, the result of careful experimental technique and long exposure times to capture the clearest possible image. The pattern visible in the photograph contained crucial information about the structure of DNA. To a trained eye, it clearly indicated that DNA had a helical or spiral structure and provided data that could be used to calculate the precise dimensions of that helix.
And this photograph was shown to Watson and Crick without Franklin's knowledge.
Yes, and this is one of the most controversial and troubling aspects of the whole story. Maurice Wilkins, Franklin's colleague at King's College, showed the photograph to James Watson without Franklin's knowledge or permission. Watson immediately recognized its significance. The information in the photograph, combined with other data that Watson and Crick had access to, proved crucial to their ability to build the correct model of DNA structure.
How did Franklin feel about working at King's College at that time?
Franklin's experience there was deeply difficult, and not only because of the incident with her photograph. The scientific culture of the time was strongly male-dominated, and King's College was a particularly difficult environment for women. Wilkins had initially misunderstood Franklin's role, treating her as an assistant rather than as an independent researcher of equal standing. The relationship between them was tense and often unpleasant. Franklin was excluded from the informal social gatherings where male scientists discussed their work. She worked largely in isolation, without the collaborative support that Watson and Crick enjoyed in Cambridge.
That sounds like a very unfair situation.
It was deeply unfair, and it had consequences that extended beyond Franklin's personal experience. Working in a more supportive environment, With better access to the work of colleagues, Franklin might have solved the structure of DNA herself before Watson and Crick did. She had much of the necessary evidence already in her data, but the barriers she faced, both social and institutional, slowed her progress in ways that her male colleagues did not experience.
Did Franklin know how important her photograph was to Watson and Crick's discovery?
No, not at the time. She was not told that her photograph had been shown to Watson. She continued her own work independently. Franklin left King's College in 1953 to work at another institution, where she went on to do important work on the structure of viruses. She died of cancer in 1958 at the age of 37, before the full significance of her contribution to the DNA discovery became widely known. She died without receiving the recognition she deserved.
And Watson, Crick, and Wilkins received the Nobel Prize.
They did, in 1962. The Nobel Prize is not awarded posthumously, so Franklin could not have received it, even if the committee had chosen to recognize her contribution. But many historians of science argue that her contribution was fundamental, and that the way it was used, without her knowledge or consent, raises serious ethical questions that the scientific community was slow to acknowledge. Today, Rosalind Franklin is increasingly recognized as a central figure in one of the greatest scientific discoveries of the 20th century.
So Watson had seen Franklin's photograph. What happened next at Cambridge?
Watson and Crick worked with extraordinary intensity in the weeks following Watson's visit to King's College. They now had several crucial pieces of information. They had the data from Franklin's photograph, which told them that DNA was helical and gave them key measurements about its dimensions. They also had access to a set of rules discovered by a biochemist named Erwin Chargaff, which turned out to be absolutely essential to solving the structure.
What were Chargaff's rules?
Chargaff had spent years carefully measuring the amounts of the 4 chemical bases that make up DNA. DNA contains 4 bases: adenine, thymine, guanine, and cytosine. What Chargaff discovered was that in any sample of DNA, the amount of adenine always equals the amount of thymine, and the amount of guanine always equals the amount of cytosine. This one-to-one pairing was a consistent pattern across all the organisms he studied.
And Watson and Crick understood what that pattern meant.
When they combined Chargaff's rules with the information from Franklin's photograph, the meaning became clear. If adenine always pairs with thymine and guanine always pairs with cytosine, then the bases must be pairing with each other across the middle of the molecule. This suggested a structure where 2 strands of DNA run alongside each other, held together by these specific base pairings in the middle.
And that is the double helix.
Exactly. Watson and Crick worked with physical models, actual metal plates and rods representing the different atoms and chemical bonds, assembling and reassembling possible structures until everything fit together correctly. The double helix model had 2 strands twisting around each other like a twisted ladder. The sides of the ladder were made of sugar and phosphate molecules. The rungs of the ladder were the paired bases: adenine paired with thymine and guanine paired with cytosine.
How did they know when they had the right model?
Everything fit. The dimensions of the model matched the measurements from Franklin's X-ray data. The base pairing rules explained Chargaff's observations, and crucially, the structure immediately suggested a mechanism for how DNA could copy itself, which was exactly what a molecule carrying genetic information needed to be able to do. When a structure solves multiple problems simultaneously and fits all the available evidence, scientists recognize it as almost certainly correct.
What was their reaction when they realized they had found it?
By all accounts, it was a moment of enormous excitement. Crick reportedly told everyone in a Cambridge pub that they had found the secret of life, which was not an exaggeration. Watson later described the feeling of seeing all the pieces fall into place as one of the most extraordinary experiences of his life. They immediately understood the magnitude of what they had found.
And then they published their findings.
Yes. On April 25, 1953, the journal Nature published a short paper by Watson and Crick describing the double helix structure of DNA. The paper was only about 900 words long, which is extremely brief for a scientific paper of such importance. But it contained one of the most famous understatements in scientific writing. Near the end, Watson and Crick wrote that the structure they had proposed suggested a possible copying mechanism for the genetic material. That quiet sentence pointed toward the entire future of molecular biology,
Why was that sentence so significant?
because it hinted at something profound. If the two strands of the double helix could separate, and each strand could then serve as a template for building a new complementary strand, then one DNA molecule could become two identical DNA molecules. This was exactly the mechanism needed for a cell to copy its genetic information before dividing. The structure of DNA contained within it the explanation of how life reproduces itself at the molecular level. That insight, contained in a single careful sentence, opened an entirely new era of biology.
And the paper in Nature also appeared alongside other papers from the King's College Group.
Yes, and this is an important detail. In the same issue of Nature, papers by Wilkins and by Franklin and her colleague Raymond Gosling were also published, presenting the X-ray evidence that supported the double helix model. So Franklin's experimental work was published and credited in the scientific literature. But the way her data had been used by Watson and Crick without her knowledge was not disclosed at the time. That part of the story only became widely known much later, largely through Watson's own memoir published in 1968, which described the events in ways that many found troubling.
The discovery changed biology completely.
It transformed biology from a largely descriptive science into a molecular and mechanistic one. Before the double helix, biologists could describe what living things did, how they grew, how they reproduced, how they responded to their environment. After the double helix, biologists could begin to explain how living things worked at the most fundamental level in terms of molecules and chemical reactions. It was one of the most significant intellectual transitions in the history of science.
So the structure of DNA was known. But how does it actually work? How does a molecule carry the instructions for building an entire living organism?
This is where the story moves from structure to function, and it is just as remarkable as the discovery of the structure itself. To understand how DNA works, we need to think about what a living organism actually needs to do. It needs to grow, to maintain itself, to respond to its environment, and to reproduce. All of these activities require proteins, the molecular machines that carry out almost every function in a living cell.
And DNA somehow contains the instructions for making those proteins.
Exactly. DNA is essentially an instruction manual written in a chemical language. The alphabet of that language has only 4 letters, the 4 bases adenine, thymine, guanine, and cytosine. But by arranging these 4 letters in different sequences, an enormous amount of information can be encoded. Just as the 26 letters of the English alphabet can be arranged to write every book in a library.
How does the cell read those instructions?
The process happens in 2 main stages, which scientists call replication and expression. Replication is how DNA copies itself. Expression is how the information in DNA is used to make proteins. Let us start with replication, because it follows directly from the double helix structure.
You mentioned that Watson and Crick hinted at a copying mechanism in their paper.
Yes. The key is the base pairing rules. Adenine always pairs with thymine, and guanine always pairs with cytosine. So if you know the sequence of bases on one strand of the double helix, you automatically know the sequence on the other strand. They are complementary to each other. When a cell needs to copy its DNA before dividing, specialized proteins unzip the double helix down the middle, separating the two strands. Each strand then serves as a template. New complementary bases are brought in and connected in sequence, following the pairing rules. The result is two complete double helices, each identical to the original.
It really is like a zipper opening and then being used to make two new zippers.
That is a very good way to think about it. And the process is remarkably accurate. The molecular machinery that copies DNA makes very few errors, approximately one mistake per billion base pairs copied. Special proteins proofread the newly copied DNA and correct most errors. This high-fidelity copying is essential because errors in DNA, called mutations, can cause cells to malfunction.
And what about the second stage? How the information in DNA is used to make proteins.
This process is more complex and involves several steps. First, a section of DNA is copied into a related molecule called messenger RNA. This process is called transcription. The messenger RNA carries the genetic information from the nucleus of the cell, where the DNA is stored, out to the rest of the cell where proteins are made.
So the messenger RNA is like a working copy of the instructions.
A very good analogy. The original DNA stays safely stored in the nucleus, like a master document kept in an archive. The messenger RNA is like a photocopy that can be taken out and used without risking damage to the original. Once the messenger RNA reaches the protein-building machinery of the cell, it is read 3 bases at a time. Each group of 3 bases, called a codon, corresponds to a specific amino acid, which is one of the building blocks of proteins. By reading the sequence of codons, the cell assembles a chain of amino acids in the correct order, folding into a specific protein.
So the genetic code is essentially a translation system between the language of DNA and the language of proteins.
Exactly. And cracking that genetic code, figuring out which codon corresponds to which amino acid, was one of the great scientific achievements of the 1960s, just years after the double helix structure was discovered. Scientists systematically worked out all 64 possible codons and the amino acids they specify. The genetic code turned out to be nearly universal, meaning that essentially the same code is used by bacteria, plants, insects, and human beings. This universality is powerful evidence that all life on Earth shares a common ancestor.
That is a remarkable finding in itself.
Deeply remarkable, and it had enormous practical implications. If all living things use the same genetic code, then it becomes possible to take a gene from one organism and express it in another. A human gene placed into a bacterium can be read and translated by the bacterial machinery, producing human protein. This is the foundation of modern biotechnology. Insulin for treating diabetes, for example, is now produced by bacteria carrying human insulin genes, a direct application of the universal genetic code.
So the discovery of DNA structure did not just explain biology. It opened up entirely new possibilities for medicine and technology.
It created an entirely new field, molecular biology, and with it a new era of medicine, agriculture, and scientific investigation. The questions that scientists could now ask, and the tools they could use to answer them, were completely transformed. Biology became a science of molecules and mechanisms rather than just of organisms and behaviors. And that transformation is still accelerating today.
So molecular biology was born from the discovery of DNA structures. What happened next in the decades that followed?
The decades following the discovery of the double helix were a period of extraordinary scientific progress. Each advance built on the previous one, accelerating the pace of discovery. Scientists developed new tools and techniques for reading, copying, and eventually manipulating DNA. The technology improved so rapidly that things which seemed completely impossible in 1953 became routine laboratory procedures within just a few decades.
What were some of the most important of those advances?
One of the most significant was the development of DNA sequencing, techniques for reading the exact sequence of bases in a DNA molecule. Early sequencing methods were slow and laborious, but the technology improved steadily, and by the 1980s, scientists began to ask a question that would have seemed absurd just a generation earlier. Could we sequence the entire human genome? Could we read out all 3 billion base pairs that make up the complete genetic instruction manual of a human being?
That sounds like an enormous undertaking.
It was one of the largest and most ambitious scientific projects ever attempted. The Human Genome Project was formally launched in 1990 as an international collaboration involving scientists from the United States, the United Kingdom, France, Germany, Japan, and China. The goal was to produce a complete and accurate sequence of the entire human genome. The project was expected to take 15 years and cost approximately $3 billion.
Did it succeed?
It succeeded, and ahead of schedule. A working draft of the human genome was announced in 2000, with the complete sequence finished in 2003, exactly 50 years after Watson and Crick published their paper describing the double helix. The announcement in 2000 was made jointly by the Public International Project and a private company called Celera Genomics, which had been racing against the public effort using faster but somewhat different methods. President Bill Clinton and British Prime Minister Tony Blair made a joint announcement calling the completion of the draft genome a landmark in human history.
What did scientists actually find when they read the human genome?
Many findings were surprising. Before the project, scientists had estimated that the human genome might contain around 100,000 genes. The actual number turned out to be far smaller, around 20,000 to 25,000 genes, not many more than a much simpler organism like a roundworm. This was humbling and fascinating. The complexity of human biology could not be explained simply by having more genes than simpler creatures. The relationships between genes and the way genes are regulated and expressed turned out to be far more important than the number of genes alone.
And what has knowing the human genome actually made possible?
It has transformed medicine in profound ways. Scientists can now look at a patient's DNA and identify variations that increase the risk of certain diseases, including various cancers, heart disease, and many inherited conditions. This kind of genetic testing allows for earlier intervention and more personalized treatment. Doctors can now sometimes choose treatments based on the specific genetic characteristics of a patient's tumor, rather than using the same treatment for everyone with the same type of cancer.
And then there is CRISPR. That seems to be something people talk about a great deal now.
CRISPR is perhaps the most significant advance in genetic technology since the discovery of the double helix itself. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is a system that allows scientists to edit DNA with extraordinary precision. It works somewhat like a molecular pair of scissors that can be directed to cut the DNA at a very specific location. Once the DNA is cut, scientists can remove, replace, or modify the genetic sequence at that point.
Where did this technology come from?
It came from studying bacteria. Scientists discovered that bacteria have a natural defense system against viruses that uses a mechanism very similar to what CRISPR does in the laboratory. Researchers, particularly Jennifer Doudna and Emmanuel Charpentier, adapted this natural bacterial system into a practical and precise gene editing tool. Their work earned them the Nobel Prize in Chemistry in 2020. The development of CRISPR was so rapid and so transformative that it caught even many scientists by surprise.
What can CRISPR actually be used for?
The potential applications are vast. In medicine, CRISPR is being tested as a treatment for genetic diseases caused by specific mutations in DNA. Sickle cell disease and certain forms of inherited blindness are among the conditions where early clinical trials have shown promising results. In agriculture, CRISPR is being used to develop crops that are more resistant to disease, drought, or pests, without the introduction of genes from other species that characterizes older forms of genetic modification.
But CRISPR also raises very serious ethical concerns, does it not?
Extremely serious ones. The technology is powerful enough to edit not only the cells of a living adult, but also the cells of an embryo at the very earliest stages of development. If the genome of an embryo is edited, those changes will be present in every cell of the resulting person, and potentially passed on to their children. This opens the possibility of what some people call designer babies, where parents could theoretically select or modify genetic traits in their children before birth.
Has that actually happened?
In 2018, a Chinese scientist named He Jiankui announced that he had created the first gene-edited human babies, twin girls whose genomes he had edited using CRISPR before they were implanted as embryos. The announcement caused an immediate and intense international reaction of shock and condemnation from the scientific community. He had acted outside established ethical guidelines and without proper oversight. He was subsequently tried and convicted in China and sentenced to prison. But the fact that it had been done at all demonstrated that the technology was real, and that the ethical questions it raised were urgent and practical, not merely theoretical.
What are the main ethical concerns about editing human embryos?
There are several. One concern is safety. Gene editing is not yet perfectly precise and can sometimes make unintended changes elsewhere in the genome. The long-term consequences of such off-target effects in a human being are unknown. Another concern is equity. If gene editing therapies become available, will they be accessible to everyone, or only to those who can afford them? A world where wealthy families can give their children genetic advantages that poor families cannot is a deeply troubling prospect.
And there are also concerns about what kinds of traits people might try to edit.
Yes, editing out a severe genetic disease is one thing, but the same technology could theoretically be used to try to enhance intelligence, physical ability, or other traits. The line between treatment and enhancement is not always clear, and different societies and individuals draw that line in different places. These are not questions that science alone can answer. They require broad social, ethical, and political debate about what kind of world we want to live in.
So the discovery of DNA structure has led us to a point where we can actually rewrite the instructions of life.
We are at the very beginning of that capability, and like every powerful technology in human history, it brings with it both extraordinary promise and serious danger. The promise is the possibility of eliminating terrible inherited diseases, of creating better treatments for cancer, of understanding the biological basis of human health in ways that were previously impossible. The danger is the possibility of misuse, of creating new forms of inequality, of making changes to the human species, whose consequences we cannot fully predict or control.
Looking at everything from the early experiments about what carries genetic information to CRISPR and gene editing today, what is the most important thing to understand about this whole story?
I think the most important thing is to appreciate both the beauty and the responsibility that come with this kind of knowledge. The discovery of the double helix was one of the most elegant scientific achievements in history. The structure Watson and Crick described is genuinely beautiful. Two strands twisting around each other, held together by specific chemical partnerships, carrying within their sequence the instructions for building every living thing on Earth. It is a profound and wonderful thing to know.
And yet the story of how it was discovered has some troubling aspects as well.
Yes, and those aspects are important to acknowledge honestly. The treatment of Rosalind Franklin, the use of her data without her knowledge or permission, the way her contribution was minimized for decades, these are real failures. They remind us that science is a human activity, conducted by human beings with all their limitations, biases, and capacity for both generosity and selfishness. Recognizing those failures is part of understanding the full story, and part of building a more fair and honest scientific culture going forward.
And now with CRISPR, we face new responsibilities that are even larger.
Much larger. When Crick and Watson discovered the structure of DNA, they could not have imagined that within 70 years, scientists would be using that knowledge to edit the genomes of living human beings. The pace of progress in this field has been extraordinary, and with that progress comes a responsibility that cannot be left to scientists alone. The decisions about how to use gene editing technology, who should have access to it, what kinds of modifications should be permitted, and what kinds should be prohibited. These are decisions for all of society to make together.
How should ordinary people engage with these questions if the science is so complex?
The science is complex, but the ethical questions at the heart of it are ones that every thoughtful person can engage with. Questions like, what does it mean to treat a disease versus enhancing a normal trait? Who should decide what is normal and what needs to be fixed? How do we ensure that powerful new technologies benefit everyone, rather than only the privileged few? These questions do not require a PhD in molecular biology to think carefully about. They require exactly the qualities that good citizens of any democratic society need: the ability to listen, to reason carefully, and to consider the interests of people beyond yourself.
It is remarkable to think that it all started with scientists asking what molecule carries hereditary information.
That is the nature of fundamental scientific discovery. You ask a question about the basic workings of nature, and the answer you find opens doors that nobody had imagined were there. The scientists who worked on DNA structure in the 1950s were trying to understand something fundamental about life. They could not have predicted where that understanding would lead. The story of DNA is not finished. The most consequential chapters may still be ahead of us. What matters is whether humanity develops the wisdom to match its expanding knowledge. Thank you for joining us today.